just like Rob, I extend my respect to Aboriginal and Torres Strait Islander peoples. Um, and he already showed, the importance of deep learning for segmenting images. And I think that, deep learning is becoming very crucial for medical imaging research. But currently, there is a big translational gap in using these models in the clinic due to the lack of generalizability. So today, I will share some of the work we have been doing in our group, aiming to increase the usability of these models by improving their generalizability. But before we get into that, generalizability can be understood as the ability of a model to perform its purpose on data from a new, independent cohort of patients. And the lack of generalizability in deep learning models can emerge from training the model on imbalanced data sets in terms of the underlying population and or in terms of different collection sites, image contrasts, and modalities. Focusing on different conditions in collection sites, different scanners and imaging protocols may lead to different image intensity distributions. And also performance deterioration of these models, which is the problem that we are trying to tackle here. So today I will cover the main line of research the main lines of research we have in our lab: test time adaptation, foundation models and active learning, and model deployment. But before we get into the nitty-gritty, I'll give you some motivation for why we care about image segmentation. Well, medical image segmentation involves dividing a medical image into multiple segments or regions of interest with similar properties, for example, similar gray level in an MR image. And here is the illustration of how we would segment the white and gray matter and the subcortical structures in the human brain. This process essentially consists of assigning a label or a class to each voxel of the MR image. After appropriate segmentation, we can extract information such as the volume of each of these regions of interest. And additionally, we can generate really beautiful surface representations of the cortical and white matter. And using this kind of data representation, we can learn about the surface area of different brain structures we are interested in. We can determine cortical thickness, essentially the distance between the gray and white matter surfaces at any location. We can find out the curvature, essentially how convex and concave any given region is. So these are just a few examples of the kind of information we can extract from different anatomical structures, but segmentation is also relevant for delineating tumoral lesions, which inform different treatment plans, such as surgery and radiotherapy, just like Rob was mentioning before. And if, like me, you are also interested in determining the functional role of different brain regions, you would also need some sort of segmentation or parcellation to delineate boundaries between brain regions, like the visual areas in the visual cortex shown here. But I guess a couple of you or most of you know that segmenting medical images take a long time, as you can see in this video here. You essentially scroll through the image, select a slice, color in different parts of the image with respect to the class they belong. You check adjacent slices to see if the segmentation makes sense, and so on. All of that assuming you have the expertise to identify the regions of interest appropriately. Of course, this is very time consuming. Sometimes we are lucky to find an automated method that works pretty well, like FreeSurfer for segmenting gray and white matter. But often we don't have that luxury. So there is a great effort in medical imaging research to leverage deep learning to automate this kind of tasks. But as I mentioned before, we often find deterioration in the performance of these models at test time. And that is because we train these deep learning models on narrow labeled data sets. And at test time, we deploy these models on different data sets, which with different spatial distributions of intensity values. And unfortunately, these models are very sensitive to these changes. So to illustrate, this problem, here we have a T2 weighted image and the corresponding segmentation generated with a pre-trained model. The segmentation model was trained on images from the same study, except the image illustrated here. And I can tell that the segmentation is pretty decent. But when we try to use this pre-trained model to predict segmentations on images from a different study, we find that the resulting segmentation is not so great. And it could be even worse if we had, T1 weighted images, for example. So there are many strategies to remediate this lack of generalizability problem. For example, we can fine-tune the model, which consists of further optimizing the parameters of the predictive model to generate predictions on a new data set. However, as a supervised approach, it requires labeled data. Can you imagine the amount of work to label data whenever you need to tune the model to a new study or a new data set? Another approach is test time adaptation, which is conceptually similar to fine-tuning. In this approach, we further optimize specific parameters of the model to generate predictions on a new dataset at test time. But instead of relying on ground truth labels to guide this optimization, we use a proxy segmentation, which could be generated with any other model or method. But the problem is, sometimes the proxy is not so great. And alternatively, we could, try to build a very robust model from scratch, like the segment anything model from Meta, which uses similar techniques seen in large language models. But to train a model like SAM, the Meta research team used as their initial database over 100,000 images with over 4 million masks. So for the medical domain, would that even be possible? And note, they trained models on natural images, 2D images. Another approach that would require less labeled data is active learning. Active learning involves using the model itself to find new samples to be manually labeled and added to the training data pool. And then we fine-tune the model with this new training data. This iterative process is done until we get a reasonable model that performs well on our target data set. And although this approach works, there is a need for labeling multiple data samples to train a reasonably performing model. As you can see, there is no free lunch. So considering the pros and cons of each of these approaches, our lab has been focusing on test time adaptation, foundation models, and active learning. So to start off, I will cover our work using test time adaptation. And a very nice and simple example of the utility of test time adaptation for medical image analysis is the work from Karani et al. Here, they start by pre-training a normalization model with a segmentation model using source data, labeled data, and specifically the output of the normalization model is the input for the segmentation model. At the same time, but separately, they train a denoising model. And to do so, they use the segmentations they already had for this data set, add a noise to it or blobs, and then they train a denoising model to remove this noise. And at test time, they combine all these models into a unique pipeline. And they freeze the parameters of the segmentation model and the denoising model while they allow the adaptation of the normalization model. So at test time, essentially, we will adapt the parameters of this model such that the prediction of the segmentation model looks like the prediction of the denoising model. So essentially, we are trying to optimize the prediction. The normalization model, in other words, would learn how to pre-process these new data sets such that the segmentation learns how to perform a good segmentation. But, sorry, although they have demonstrated the effectiveness of this approach using data from common medical imaging challenges, it is hard to tell how effective that is when we move to clinical data, where there are data from patients' populations, from diverse acquisition parameters, and with artifacts. To answer this question and at the same time generate segmentations with clinical relevance, I've adapted their approach to something somewhat simpler by using an atlas as our proxy as opposed to having this extra denoising model. So specifically, we tested if it would be possible to use this approach to segment tongue muscles from healthy controls and motor neuron disease patients from several data sets. My collaborator Tom Shaw has been putting a great effort in acquiring data from MND patients from several studies, including Belong and EAT. So we combined his needs to get segmentation for this clinically relevant data with my work to propose more robust and efficient deep learning models for medical image segmentation. In our approach, we start by pre-training the normalization and segmentation model using T2 weighted images from the Belong study, for which we had roughly 15 manually labeled data images. So it's not a lot. And at test time, we adapted the weights of the normalization model to images of different studies while keeping the segmentation model fixed. So the parameters of the segmentation model is frozen, while the parameters of the normalization model is allowed to be adapted. We use the registered atlas as a proxy segmentation to guide this optimization. So essentially, we compute how different is the output of the segmentation model in comparison to the atlas. And all the images were registered to the atlas space. And importantly, this normalization model is adapted separately for each participant. Here we can see on the left-hand side the atlas registered to a patient image. It's a pretty reasonable registration. And on the middle, we have the prediction generated with the pre-trained model. As I said, these pre-trained models, when we try to generalize to different data sets, we don't expect good performance. And we can see clearly here that the segmentation is not great. It's pretty rubbish. But then after we adapt the normalization model using the atlas as our proxy to guide this optimization process, we find a really nice segmentation now. And when we do the same process for T1 weighted images, we see that this approach also works. But what if the atlas is not well registered to the underlying image, like the example we have here? Well, when we run adaptation of the normalization model, this is the kind of segmentation we get out of it, of the entire pipeline. And we can see that we kind of guide some sort of we get some sort of overfitting. So the final segmentation is slightly shifted to the right, just like the atlas. So in summary, we find that test time adaptation is an effective method for adapting a pre-trained segmentation model to images of, different contrast and collection sites, but the final segmentation are only as good as the proxy. Another alternative for test time adaptation is using the predictions generated with the pre-trained model as our proxy instead of the atlas. And I've been exploring that in collaboration with Saskia Bowman and Marshall Shu to better understand the organization and function of the brain arterial vasculature. Saskia has been doing an amazing work acquiring magnetic resonance angiography data at a mesoscopic scale, and segmenting this kind of data has its own challenges. It is time consuming, it is noisier than macroscopic scale data, there is a lower vessel to background contrast. And aiming to address the need for the segmentation of mesoscale vessels, in earlier work, Saskia and team have found that by using deep learning in a clever data augmentation that leverages the self-similarity of large and small vessels, it is possible to boost rough segmentations and have improved sensitivity to small vessels. So this clever data augmentation consists of randomly cropping the input data at different locations with different sizes, and then resizing the patches to a fixed dimension. In practice, this is equivalent to zooming in and out for patches smaller or larger than the final dimension size. To determine whether test arm adaptation would be an effective alternative to improve the performance of pre-trained models for vessel segmentation, we start by training a segmentation model using the Ribeiro challenge data. The challenge provides a public data set containing magnetic resonance angiography time of flight data at an isotropic resolution of 300 micrometer and a set of different segmentations. We use the segmentation generated with OMLAD, an automatic method, and you can notice that the segmentation is very coarse. We are missing a bunch of like the smallest vessels. And for test time adaptation, we actually adapt the weights of the pre-trained segmentation model for a few training epochs. And we use as our proxy the segmentation generated with the pre-trained model before adaptation. And here we have the ground truth segmentation for a data sample from the challenge, but that was not used for training the model itself. And this is the initial segmentation we get out of our pre-trained model, okay-ish. And this is the segmentation we get after test time adaptation using this initial segmentation as our proxy. We can note that we were able to capture more of the small vessels, effectively boosting the initial segmentation, which is pretty cool. So here is the ground truth, and you can see that, you know, the initial segmentation is a bit discontinuous, and we improved the segmentation here. So we're really happy about it, and we are actually preparing a submission for the Journal of Open Source Software. But all code is already available on GitHub for you. So if you want to, you know, segment some vessels, feel free to use it. Another active topic in our lab is the use of foundation models to reduce model development and training time. One example of foundation model for image segmentation is SAM, the Segment Anything model. And this is a very impressive model, and it is a universal segmenter. It means that it can segment any object in natural images, pretty much. And this model consists of three components: the prompt encoder, which generates a prompt embedding, an image encoder, which generates an image embedding, and a mask decoder that generates segmentations. When building SAM, the research team had three requirements, essentially supporting flexible prompts. So you can actually use points, bounding box, and mask as prompts to guide the segmentation of your image. The model should compute masks in real time to allow interactive use. And especially, we can use the same image embedding with different prompts, which allows for interactive use, and we can actually optimize the segmentation generated with the model by adding more prompts as well. And also, the model should be ambiguity aware. So SAM is actually able to predict three different masks with different level of granularity from a single prompt. So for example, here we have this point on essentially indicating for this lady here, and in the first output of SAM we have the model segmenting the whole person. In the second output we have the model segmenting just the backpack, and in the third output we have the model segmenting just the pocket of the backpack. But SAM was not trained on medical images, just natural scenes, and it doesn't work out of the box for our purpose. So another group actually fine-tuned SAM using a large amount of publicly available labeled medical images, and they use several medical images with different anatomical structures, different modalities, and pathologies. And they actually demonstrate that this fine-tuned model performs better than SAM for medical image segmentation. However, you can only get one anatomical structure at a time. And unfortunately, Xincheng actually tested MedSam, and we found out that it doesn't generalize well either. So Xincheng, a PhD in our lab, is actually using concepts from active learning literature to determine an effective way to tune SAM with the least amount of data in a case by case. So we can leverage the learned knowledge from this pre-trained model at the same time when we adapt it minimally to new tasks. So the idea is really to find what is the minimal number of labeled samples required to tune SAM for organ segmentation, for example. Specifically, in the active learning framework, we use the initial state of the model to generate segmentations, and then we estimate how uncertain the model is in predicting that segmentation. We then rank the labeled images based on the uncertainty metric, and we select the top ranking image to be manually annotated. Then the user, fix the segmentation and add that new annotated sample to the training data set, to the training pool. And then with that new, training data set, we fine-tune the model and we perform this whole process again. So this is a iterative process, and we grow our training data until we get a reasonably performing model. So the assumption of this idea is that by adding these images that the model is most asserted about, we are increasing the diversity of the training data. So actually, Xincheng has already done something similar, but using conventional UNet, and with just over 50 labeled images, she was able to train a reasonable model to segment kidney, and applied to over 900 images, and the segmentations were very nice. So our expectation is that with SAM, we would need even less images to train a good performing model. And another line of development in our lab is model deployment. And there are many challenges when trying to get these models into production. For example, model generalizability is a big issue, and this is what I've been, discussing about. But I actually have shown a couple of strategies that we have been using to minimize this problem. But apart from that, we can also work on having more flexible code for training study-specific models. So actually, other people can use our framework as well. But flexible code doesn't equal duplicated code. With minor edits, we have to work towards more general functions and functionalities, but this is a lesson I learned during my first year of postdoc. In our lab, there is a strong culture to streamline code and build tests continuously, which was a major paradigm shift for me. And because of that, I was able to develop a command line interface for a deep learning model I developed during my PhD to predict retinotopic maps in the human visual cortex. So if you're interested in generating retinotopic maps, please check out the toolbox as well. And another major challenge to consider is code usability. We often see researchers sharing their code on GitHub and pre-trained models wait somewhere else, but it is often difficult to deploy them locally because of dependency issues. In that sense, software containers greatly reduce this complexity as it allows for packaging of software along with software dependencies all together in a lightweight, isolated container. And thanks to Stefan and the NeuroDesk team, the complexity of using and building software containers for neuroimaging research has been greatly reduced. NeuroDesk greatly facilitates the translation of our research into practical use by other researchers, as we don't need to worry about software installation across multiple computing environments or the lack of reproducibility of computation analysis. For example, on NeuroDesk, there are many software containers with the most used neuroimaging software. Besides, you can easily contribute with a container for your application. Of course, NeuroDesk is tailored to help neuroimaging researchers, but our lab is also working on a way to translate medical imaging research to the clinic by establishing a framework to deploy pre-trained segmentation models on the browser. And here we have a demonstration of the tool TUI has been developing. TUI is a PhD student in our lab, and she's currently developing a framework to deploy pre-trained models on the browser, where a potential user could just drag and drop their data. And here we have SAM, the segment anything model, deployed on the browser. And with a few prompts, we can see that it is possible to guide and improve the generated segmentation. So in summary, I've discussed how we can use test time adaptation to improve the performance of pre-trained models on out of distribution data, virtually recycling these models and obtaining segmentations in a faster fashion. And I also discussed the potential of foundation models for medical image analysis and how we could potentially combine these models with active learning. And I also discussed how we have been addressing the challenges in bringing models into production, and hopefully there were a couple of valuable tips for you. I'd like to acknowledge our group leader, Stefan Bowman, he's around, my collaborators and lab mates, and of course, all the funding that we have been, accumulating over the years. And yes, thank you for your attention.